Hi, everyone. Welcome to OsteoBites. We'll give a few seconds for everyone to get settled in our virtual room. Thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Troy McEachron from the Pediatric Oncology Branch of the National Cancer Institute, where he leads the Integrated Solid Tumor Biology section. And he joins us today to discuss the molecular and cellular deconstruction of metastatic osteosarcoma. Thank you so much, Dr. McEachron, for joining us on OsteoBites today. We are thrilled to have you. And thank you also to our panelists, Walker and Daniel, for joining us. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom. You'll see that in the bottom menu of your Zoom panel, and we will share those with Dr. McEachron after his presentation. A little bit more about our guest today. Dr. McEachron earned his doctorate in Molecular and Cellular Pathology from the University of North Carolina at Chapel Hill in 2011, and he completed his postdoctoral fellowships at St. Jude Children's Research Hospital and the Translational Genomics Research Institute. In 2016, he joined the faculty of the Keck School of Medicine at the University of Southern California as an assistant professor in the Department of Translational Genomics and the Department of Pediatrics. He then joined the Pediatric Oncology Branch of the National Cancer Institute in 2021, where he leads the Integrated Solid Tumor Biology section. And the major focus of his laboratory is to molecularly dissect the microenvironment of pediatric metastatic osteosarcoma to better understand the biology of metastatic disease and identify therapeutically actionable targets. Thank you so much for joining us today, Dr. McEachron. We are very much forward to your talk. Just a couple of quick announcements before we get started. Some exciting ones today. We have our 2025 Outsmarting Osteosarcoma Research Grant Letter of Intent submission period opening tomorrow, September 27th. Letters of intent are due in Proposal Central by October 25th at 11:59 p.m. And this is actually a new thing that we started this year, adding a letter of intent step to our application process based on feedback actually from applicants. And so we're excited to do that, but it means we're getting an earlier start this year. So put that on your calendar. I hope people are planning to submit this year, October 25th, 11:59 p.m. I'm going to put a link in the chat where you can find more information. You can access the program guidelines, read the eligibility requirements, also just see what's required for the LOI, and then also access the Proposal Central link. And then super exciting information, save the date. You heard it here first. Factor 2025 is going to be June 26th to 28th at the Grand America Hotel in Salt Lake City, and registration will open for that in February of 2025. So Dr. McEachron, I hope you're blocking that out on your calendar. Maybe you can plan a little family summer vacay around Salt Lake City. Should be fun.
Yeah.
And we are going to be accepting abstracts for Factor. Abstract submission will open on October 7th. And they are due January 10th, 2025. So good amount of time to submit those, just to kind of everyone start thinking about that. And we hope to see lots of abstracts for that as well. Lastly, clinicians and researchers with an interest in osteosarcoma are invited to join Turbo, which is our virtual tumor review board for osteosarcoma. It's an educational forum where clinicians discuss complex cases. And it's also a great way for researchers who don't see patients to learn about real-world treatment challenges. Our next meeting is November 6th at 5 p.m. Eastern. And if you are interested in presenting a case or you would just like to attend, you can email me at christina@mibagents.org and I will make sure you get on the calendar invite. All right, that is it for me, but lots of exciting stuff happening this fall. How about Daniel? Could you go ahead and introduce yourself?
Hi everyone, I'm Daniel. I had osteosarcoma when I was in middle school, and I had a distraction osteogenesis surgery, and I now have a rotationplasty, and I'm now in senior year of high school.
And Walker.
Oh, sorry. Hi, my name's Walker. I'm part of the junior advisory board, have been for the last few years. Like Daniel, I'm also an osteo survivor. I'm really excited to hear more about today's topic. Daniel and I both actually had the opportunity to go to Dr. McEachron's lab in March, and I'm really excited to hear more about his research and what they're doing in the lab. So Dr. McEachron, you can take it from there.
Great. I will share screen. All right. All right. Hopefully you can see this well. Okay, great. Well, thank you so much for the opportunity to present. So today I'll talk to you about the molecular and cellular deconstruction of metastatic osteosarcoma, or in short, kind of what are we doing in our lab? So as a government employee, the mandatory slides. So most people here don't need an introduction to osteosarcoma, but just in the event that you do, I'll try to kind of go over this few things really quickly. So osteosarcoma is a genomically complex disease, as you can see here from this circles plot. This is whole genome sequencing from a patient with metastatic osteosarcoma. And what we can see is that there's a bunch of intra and intertumoral translocations. So essentially all of these lines, let me grab my laser pointer. All of these lines that are actually inside the circle here are not supposed to be there. And when we look at osteosarcoma in the context of a variety of other pediatric malignancies, we can see that these are highly complex tumors. And these tumors don't necessarily, the genomic complexity doesn't necessarily equate to the expression of neoantigens. But we also know that osteosarcoma is driven by numerous copy number alterations. So rather than having point mutations, we have a variety of copy number alterations. These include gains and losses, so extra pieces or deletions of chromosomes, whole chromosomes, chromosome pieces, so on and so forth. So it's really, really complex. And from the genomic standpoint, these are kind of N of one diseases because these are the majority of osteosarcomas are so complex and present a really unique genomic landscape in each individual tumor. So from the kind of current status of osteosarcoma, survival rates have not really improved in over 40 years. And what this means is that patients with metastatic disease actually have an inferior survival. And this is actually an unmet medical need. So this is where my laboratory hangs our hat. We really want to understand what is going on in the metastatic context. When we think about clinical trials, there have been a variety of agents that have been tried, including a variety of different immunotherapeutic approaches, and these have provided little benefit to patients diagnosed with osteosarcoma. So when you put that together, what we're really interested in doing is trying to understand the context of metastatic osteosarcoma and why some of these therapeutic approaches have actually not worked for patients battling this disease. So rather than focusing on the kind of genomic complexity of the disease, which has been the vast majority of the osteosarcoma research community up until recent years, we really want to understand the tumor microenvironment. So there's a lot of talk about the tumor microenvironment, and most often people kind of conflate tumor microenvironment and the immune system, but it's the tumor microenvironment is much more than a bunch of tumor cells and immune cells. As you can see here, from this graphic, you have extracellular matrix, you have a variety of different chemokines, cytokines that are chemically modified, vasculature, which has its own function, nutritional gradients, metabolic gradients, and then immune cells. And then, so all of these things kind of come together, and they're not really put together haphazardly. They actually do have a structure and a function. So the tumor microenvironment, we can think of it as kind of this co-opted microenvironment. So there's kind of this kind of tug of war, right? Where the tumor is wanting to do one thing, the body's wanting to do another thing, and they're trying to really figure out who's going to win that battle. So there's this evolving state of tension as it pertains to the microenvironment. And the cellular molecular composition of the microenvironment is really going to be dependent on the tissue type and tissue site. So each tissue is made of a variety of different cells. Each one of those cells is responsible for maintaining homeostasis of that tissue. So the microenvironment in tissue A and tissue B are not really going to be the same because the cells that comprise tissue A and tissue B are also not going to be the same. And that means that the resident cells and the extracellular factors in each tissue will actually orchestrate these site-specific responses. So meaning that the metastatic lesion in site A versus metastatic lesion in site B, the molecular and cellular mechanisms that comprise the function of that metastatic site will likely be different due to the fact that they are comprised of different tissue, different cells.
Ashley, can I just clarify that point? When you say different sites, does that mean like bone versus lung, for example, or does that mean that it could even be two different bone sites, like could
So
be
this is actually, that's a wonderful question. And I'm actually going to get to that right now. So typically, yes, we're thinking about different anatomical locations. So bone versus lung, for instance. But one can actually start asking a slightly different question, right? So when we think about the bone, we think about, okay, well, it's bone. There's not that much there. But the axial skeleton and the appendicular skeleton, axial being the spine, rib cage, all that other stuff, and then the appendicular skeleton being the long bones in the arms. The composition of the bone marrow, the structure of the bones, bone remodeling, turnover, they're actually different in different sites. So you could even say within the bone, if you have let's say a tumor in the pelvic girdle versus a tumor in the distal femur versus a jaw osteosarcoma. While we don't necessarily know this to be true for sure, and there needs to be more research going into this, but there's likely going to be different microenvironmental factors that will actually have the tumor, the molecular and cellular features of those tumors will likely be different. But we need to do more research to figure that out, and that's one of the things we're really interested in doing. Does that answer your question?
Yes, thank you.
Okay, perfect. Oops, here we go. So this is what, an osteosarcoma looks like. So when we think about the tumor microenvironment, what we can see here is that we have malignant osteoid, malignant chondroid, blood vessels, red blood cells, and then a variety of other cells kind of all put together. So we need to try to figure out like what's going on and what are we doing with this. One of the things that's really unique about osteosarcoma is that, there are more dogs that get osteosarcoma than there are humans. So that allows us to actually take a comparative oncology approach to study the tumor microenvironment. And as you can see from these images, the presentation of metastatic disease is very similar in canines and humans, as you can see here from these radiographs and this imaging here. And then when you actually look at the tumors under a microscope, the cellular composition, the microenvironmental factors, a lot of these things are actually well conserved between human and canine. So it allows us to really utilize companion animals, utilize pet dogs that naturally develop osteosarcoma to learn a lot more about that disease and then apply that to humans. Or on the other side of the coin, what we do in humans, we can then try to validate in a larger cohort of canine specimens to make sure that the conclusions that we're drawing are statistically valid utilizing a larger sample set. So we study pediatric osteosarcoma. So why is that important? One, children are not small adults. And when it comes to AYAs, that's a whole different story. That's a whole nother can of worms because you're kind of caught in the middle for a variety of different factors. But we do know that the age impacts the local and systemic microenvironments. With sarcomas in general, you can actually have the same diagnosis across multiple distinct anatomical locations. And we do need a more sophisticated molecular and cellular stratification criteria to see if we can bin disease by some of these characteristics to offer different therapeutic or prognostic variables associated with that disease. So let's talk about the impact of age. So the thymus shrinks as we age, and that's where we get all our T cells from. So this is really just goes to show that as we get older, there is a shift in our immune system. And at the same time, we have this thing called inflammaging. So the baseline levels of inflammation that we have actually increases as we get older. Now, why is this actually relevant? And I'll show you why. Osteosarcoma usually peaks in this adolescent age range, but can actually creep back up in adults. And here's a paper, beautiful paper, looking at the immunogenomic landscape of osteosarcoma. And what I really want you to pay attention to is this heat map in which they looked at bulk RNA sequencing data and then clustered it and inferred a variety of different immune cell types to just try to get an understanding of what the immune microenvironment looks like. When you parse this out, and you look at clusters 1 and 2, which are related, versus cluster 3, which is actually kind of distally related, there's no difference in vital status between cluster 1 and 2 and cluster 3. There's no difference in disease status, but there is a difference in age, where cluster 3 seems to be older patients, whereas clusters 1 and 2 seem to be younger patients. And then when we look at this heat map, what we can see is that cluster 3 has more red, which means that that's a much more inflamed tumor. That's a much more immunologically active tumor versus clusters 1 and 2, which are less immunologically active. So age might actually have an influence on on the cellular and molecular processes going on within the microenvironment. And as we start thinking about immunotherapy and all these things, this asks the question, should age be a variable that we should be considering? Other things, the impact of age on the non-immune microenvironment. So the extracellular matrix supports a variety of tissue structures, and this integrity decreases with age. And if you watch any bit of trashy TV like me and my wife do, you're inundated with these commercials saying hyaluronic acid and collagen creams and all these things. It's the fountain of youth. But they're not actually lying, right? Like this is actually, there's some science backed in this, not really giving credence to the cosmetic industry, but there are these factors actually are associated with the aging process, and these factors actually break down with age. And this is important because the extracellular matrix is a really, really important part of osteosarcoma. So we wanted to ask a very basic question a few years ago. What are the molecular and cellular features of pediatric osteosarcomas? So what we did is we did gene expression profiling in primary disease in the bones versus metastatic disease in the lung. And really what this is showing is that there are two very different gene expression patterns or gene expression profiles between lung metastases and primary, which is to be expected because, again, these microenvironments are going to be tissue specific. So you have tumors growing in the bone versus tumors growing in the lung. So, but this was a very interesting study, which led to a variety of hypotheses. One of the things that we noticed is that lymphocytes are actually excluded from the metastatic osteosarcoma. So essentially what this means is that when we look at the actual tissues, we can see these lymphocytes. So these are T cells stained with CD3 epsilon. They're in the non-tumor region of this metastatic lesion, but never actually make it into the core. So this white line delineates the lung parenchyma from the actual tumor core. These T cells are in the lung parenchyma, but really never make it into the tumor core. Whereas in primary osteosarcoma, you can actually see T cells kind of scattered throughout. One asks the question, well, why does this matter? Well, immunotherapy hasn't really been effective for patients with osteosarcoma, and immunotherapy is predicated on the basis of having a pre-existing yet suppressed immune response, right? So in the context of osteosarcoma, if those lymphocytes actually aren't engaging the tumor and inside the tumor tissue, it kind of stands to reason that immunotherapy wouldn't necessarily be all that effective. And John Liggand has also seen this in a paper that came out after ours. So essentially, what do we do with this? We know that osteosarcomas are relatively rare tumors, right? So how are we going to be able to access metastatic osteosarcoma and try to understand what's going on, especially with the murine models that we have right now? While all models are useful, no model truly recapitulates human disease, and that's especially true for osteosarcoma, where there's been a lot of effort and a lot of progress in making these models, but they're still not necessarily representative of what we're seeing with patients. So we decided to take, when I came to the NIH, we spent a decent amount of time trying to understand how to actually tackle this. So we decided to come up with a multi-omic profiling approach that will allow us to maximize the utility of limited patient specimens. So essentially what we have is that we obtain metastatic osteosarcoma specimens. So if we're taking frozen specimens, for instance, we will extract nuclei and do single nuclei multiome sequencing. So that allows us to sequence RNA and assess gene expression and chromatin occupancy from an individual cell. And then at the same time, we'll actually use the remainder of that specimen for bulk proteomics, and we'll do either phosphoproteomics, total proteomics, or cytokine arrays. FFP is a lot more ubiquitous, so we'll get these FFPE specimens and utilize that for spatial profiling. So we'll either utilize spatial proteomics or spatial transcriptional profiling, and I'll just really focus on these two aspects today. But we do have some really interesting data from our multiome and bulk proteomics data that we're currently exploring and trying to understand. So our first foray into this was utilizing this nanostring approach. So essentially, here is a metastatic osteosarcoma lesion from a patient, and we just decided to kind of grid across it. So we created this grid, and in each one of these boxes, we actually collect gene expression profiling. So we assess the expression of 18,000 genes in each one of these individual squares. And what we realized is, our design is to kind of make this concentric grid. So we have different layers, an outer layer working to the inner layer. So the outer layer is layer 1, and going inwards, layer 2, 3, 4, 5, and 6, just to kind of get a regional idea of what's going on. When we did the gene expression profiling, one of the things we saw is that the outer layers kind of clustered together and the inner layers clustered together. What that means is that the gene expression profiles in the outer layers are very similar. The gene expression profiles in the inner layers are very similar, but the inner layers and the outer layers are very much different from each other. So we thought that that was really cool. And this is just to show that we kind of had some gradient effects, but that's really not important because I'll show you what that looks like here. So when we look at certain lineage cell type markers or lineage markers, we can kind of see this gradient effect in real life. So these boxes, so SFTPA2, EMP, PIGR, these are associated with lung epithelium, and we can see that they're more highly expressed in the outside versus the inside. When we look at fibroblasts, we can see slight gradient effect. PTPRC is like a pan lymphocyte marker. Again, if we go back to our previous data where we see that the lymphocytes are this is a pan leukocyte marker, so all white blood cells, immune cells. When we go back to our previous data, you can see that the lymphocytes are on the outside of the tumor and then not really on the inside. And this is exactly what we see here, high expression on the outside. And then as we get closer into more towards the inside of the core of the tumor, we see decreased expression. CDH5, von Willebrand's factor, these are markers of the vasculature, but Vimentin, ALPL, CD276, these are actually markers of mesenchymal and osteosarcoma cells. And as one would expect, they are very low in the outside of the tumors and actually increase as you move inwards towards the tumor core. When we look at PTPRC, again, we can see kind of the usual characters, CD14, CD163, CD68. So these are all different immune markers and marking different immune subsets. And we could see, again, they're higher on the outside or in the periphery, and that expression actually decreases as you move inwards towards the core. So we can kind of characterize it as in the periphery, you have many more immune cells, many more immune checkpoints and inflammatory mediators, and that decreases as you actually get into the core of the tumor. Conversely, in the periphery, you have very little ECM, very little TGF-beta and CD276, which is a marker of osteosarcoma and also an immune checkpoint. But that's very low in the periphery and actually increases as you get to the tumor core. So this really does set up this hypothesis that the osteosarcoma pulmonary metastases have these regionally distinct microenvironments. So we're really interested in trying to understand what those different regionally distinct microenvironments are and what they're doing. So we utilized kind of a lower profile or a lower resolution spatial profiling platform to kind of generate that hypothesis. But now we wanted to go forward and use a much more higher resolution. So we utilize this Visium platform. So essentially what ends up happening is that you have a slide and you have this capture area. So it's 11 millimeters by 11 millimeters. So just over 1 centimeter by 1 centimeter square. You put your tissue onto that little capture area, and that allows you to actually generate 14,000 data points. So in each one of those data points, you have the opportunity to sequence the entire coding transcriptome. So if you say 18,000 genes per spot times, and the 18,000 referring to the number of genes in the coding transcriptome, 18,000 genes per spot times 14,000 spots per slide, that allows you to get a lot of data. And I'm not that great at math, so I'm not going to pretend to be able to do that multiplication really quickly, but it's a lot of data, right? So that's kind of the approach that we took here. And so each one of those spots is 55 microns in diameter. So it's really, really, really small spots. So essentially what that does, that takes us from here to something like this, right? Where each spot can contain 1 cell, 2 cells, 10 cells, or no cells. So when we look at that, so we did this on metastatic osteosarcoma specimens. So these are all pulmonary mets, 5 of which are osteoblastic histology, 1 chondroblastic, 7 of which are osteoblastic histology, 1 chondroblastic, 1 fibroblastic. These 7 are actually from the lung parenchyma, and then these 2 are from the hilar lymph node. So when we take this data and then we do our fancy profiling, one of the things we can do is project this as a UMAP. This is what people are really used to seeing when they look at single cell sequencing. So each dot here represents one of those individual capture spots, right? So we're looking at the expression of genes and just kind of clustering them to see which dots are more transcriptionally similar than the others. And what we can see here is that there's a structure. All of the everything outlined in black is from the intratumoral microenvironment or within the tumor core. And we can see kind of consistently that the tumor core, the dots from the tumor core, the spots from the tumor core kind of segregate away from those that are actually not in the tumor core. So when you take this UMAP, you combine it with the histology, that gives you something like this, where now we can take each one of these dots, figure out where in the tissue that dot actually came from, and that allows us to build, quote unquote, these spatial maps. So that can tell you where each one of these dots lives in this tissue. And each dot is representing, up to 18,000 genes. So when you actually look at the data that way, you can see that we've essentially taken this UMAP, combined it with the histology, projected it back, and now we can start revealing structure. And one of the things that we were actually able to realize is that there is still this kind of almost these concentric circles where you have the lung parenchyma followed by another layer, followed by another layer, and it kind of, as you go work from the periphery in, there are these different layers that we're actually seeing really well exemplified here and here. So we thought, okay, this is really, really interesting. So let's kind of dig into this just a little bit more. One of the things that we thought was really, really cool is that we were able to, utilizing this approach, we were able to see something that we wouldn't have been able to see underneath the microscope. So we took this fibroblastic osteosarcoma specimen. Pathologically, all you need is the evidence of malignant osteoid to be diagnosed as or classified as osteosarcoma. So when you look at this tissue, the pathologist annotated it as this entire thing is tumor. Here's a little bit of this lacy material is the lung parenchyma. The rest of this is tumor. But then taking our approach, we're saying, well, yes, this might look like tumor, but there's actually a lot more going on in this tissue than what we'd ever be able to see underneath the microscope. So been looking at it, we can see that there's different populations based off the transcriptional profile. It's not just, hey, this is one massive tumor. There's a lot more going on here. There's a lot more action here than we would have initially anticipated just based off of histology. And then when we look at it and assign some gene signatures, do some clustering, one of the things that we can see is that there's a region of this tumor that's highly osteosarcoma, and then there's regions of the tumor that's actually a lot less osteosarcoma-like, and it has many more immune cells. And we did a correlation analysis, and what this correlation analysis did is say, essentially what it says is that, the portions of the tumor that are osteosarcoma are actually negatively correlated with plasma cells, CD8 cells, so on and so forth. So, and you can kind of see that typified here. So what that's saying is that even in the context of fibroblastic osteosarcoma, where we're not by microscopy, we're just basically seeing a solid sheet of tumor cells, there still is this structure and there still is this lymphocyte exclusion that we saw in the kind of osteoblastic tissues, but it's a little bit more nuanced. So essentially utilizing this spatial profiling approach, we're still able to reveal molecular and cellular structures of these tissues that we would have never been able to see or appreciate utilizing just basic H&E staining and histopathological techniques. So going back to this kind of what we've seen previously and what John Ligon's paper showed, that this peritumoral accumulation of lymphocytes, we see the same thing. So here's just an osteosarcoma signature. Effectively, it just denotes the tumor core from the periphery, just for the sake of visualization. And one of the things you can see is that B cells, plasma cells, CD4, CD8, and NK cells, they're really not within the core of these tumors. They're kind of decorating the outside of the tumors. what we thought was really, really cool and really interesting is when we actually went to the lymph nodes. The lymph nodes are just chock full of immune cells. That's what they do. They're lymphoid organs. The fact that the regions of the lymph node that are high tumor were still able to exclude lymphocytes. So essentially what you have is a tumor growing in a lymph node. So there should be plenty of immune cells there. But within the tumor core of that lymph node, there's an absence of lymphocytes. So what that's saying is that there's something intrinsic to the osteosarcoma cells, that microenvironment, that intratumoral microenvironment that they're creating is going to exclude immune cells, regardless of where they are. Whether you're in the lung parenchyma or if you're even inside a lymph node, there's still something really special about that osteosarcoma, that intratumoral microenvironment that's saying, okay, lymphocytes, you need to get out. We're going to occupy this space and really not let you in. When we do see the few lymphocytes around, and this took quite a few specimens to actually find a cluster of lymphocytes, but when we actually do see lymphocytes, one of the things we're able to realize is that the intratumoral lymphocytes, the lymphocytes that do for some reason are able to make it into the core of these tumors, they're actually exhausted, meaning that they're functionally inert. So even though they might be there, they're not really doing much of anything. So we said, okay, this is great. We can see all of this at the RNA level, but can we see it at the protein level? Because actually, when we're thinking about drugging things and therapeutics, we're really not going after RNA, we're going after the protein. So we want to make sure that what we see at the RNA level, we can also see at the protein level. So we took a slightly different approach where we utilized imaging mass cytometry. So essentially, all this does is allow you to perform mass spectrometry on tissues. So essentially, we have a tissue, we have about 50 different antibodies, each one conjugated to a heavy metal. You put it on this fancy machine, it has a laser that scans back and forth, and anytime it hits an antibody that has a heavy metal, it ionizes it, and then it captures the mass to charge ratio, and then it records the XY position, essentially making a map of where these proteins actually are. When you take that, this is kind of what it looks like. Everything in red is a nucleus. Then all the white outlines, or we segmented this image into individual cells. So we go from nuclei to cells. Then you can create a kind of matrix to understand who's doing what. And then you could actually project that back onto the tissue to say, okay, well, now we have these individual clusters. We've been able to essentially immunophenotype this tumor. And here we used 32 or 37 different antibodies. And what this says is that, okay, we can identify different clusters. So there are cells that express different combinations of these antibodies. We can characterize them and say, okay, now where do these cells actually live in this tissue? And then we could do network analysis to then start asking questions, how similar or different are these regions of the tumor? So essentially, we're just making maps of the tumor at this point at the protein level versus the RNA level. When we do that, we can see, again, we still have this peritumoral accumulation of lymphocytes. So this is the imaging mass cytometry data. And again, you can see that within the core, we're really not seeing too terribly many lymphocytes in these osteoblastic specimens. But in this fibroblastic osteosarcoma specimens, we actually are seeing infiltration of lymphocytes, which is kind of interesting. We can take that data, do some more really fancy math, make all these UMaps and all this other really fun stuff. But essentially what this allows us to do is, again, really do dimensional reduction analysis and ask the question, what cells are expressing what types of protein and how do they actually cluster together? And then once we identify how they cluster together and what their relationships are, we can project them back onto these maps. One of the cool things that we saw is that we have these peritumoral B cells and these CD8 cytotoxic CD8 lymphocytes, and they're really next to each other. They kind of occupy, they butt up against each other in this peritumoral region, and that's really interesting because there's this concept of regulatory B cells that will actually inhibit cytotoxic function. And from previous data, what we see is that B cells in the metastases are really associated with immunosuppressive IL-10, regulatory T cells, exhausted CD8 cells, and immune checkpoints. So there's a correlation between B cells and immunosuppression in metastatic osteosarcoma that's not really present in primary osteosarcoma. So we thought that was really cool. And the fact that we have these B cells butting up against these cytotoxic lymphocytes in the periphery, we asked the question, might that be doing something? So that's something that we're really interested in trying to explore a little bit more. One of the next things we see is this kind of foreign body response. So in the periphery, we see these are multinucleated giant cells, and those are only there in the context of a foreign body response. And what a foreign body response is your body has something that it can't clear. And it's really irritating, and your body doesn't know what to do with it. So instead of trying to clear it, it says, okay, you know what? I'm sick and tired of trying to clear you. I'm actually just going to encapsulate you. I'm just going to kind of like put you in timeout, right? So I'm going to put you in timeout, encapsulate you so you can't do anything else to us anymore. We're just going to make you invisible. Now, if you're a tumor, the best thing that you could possibly do is say, yes, please put me in timeout, make me invisible. I don't want to be bothered by you. I don't want to be bothered by you trying to attack me. So it's actually beneficial for a tumor to actually be encapsulated and effectively made invisible to the immune system. And that's kind of what this foreign body response does. So we think that the tumors elicit this type of foreign body response to actually evade the immune system by just kind of being put in timeout. And we have a lot of evidence to suggest that. So we have this is the kind of workflow or what the foreign body response looks like. And evidence is we have these foreign body giant cells that we can see here. We have macrophages and myofibroblasts, so CD68 staining macrophages, smooth muscle actin staining myofibroblasts. We have capsular contracture, so this kind of fibrocollagenous capsule, and we can see that with trichrome staining, where you have this capsule kind of encompassing and encircling these metastatic lesions, and molecularly, we can see evidence of that here as well. So thus far, we have a variety of conclusions that I'll just go through quickly. These are structured tissues. Lymphocytes are excluded. Lymphocytes that are there tend to be exhausted. We have these tumor-associated B cells, and we're not sure what they're doing. And then we think that this metastatic disease elicits a foreign body-like response. So then we want to dig in a little bit further and ask questions about kind of cell-cell communication in different regions of the tumor. And essentially what we have here is a heat map that kind of we've aggregated all of our specimens together, the different spatial clusters associated with those specimens, and asked a very simple question: how related or different is the intratumoral versus extratumoral signaling pathways? And we can see that they're very, very different, right? So the intratumoral signaling is over here, extratumoral signaling is over here. What that says is that there's different communication mechanisms utilized within the tumor and outside the tumor. They're not the same. And when we can kind of look at this a slightly different way and do a volcano plot and say, we can see here that in the extratumoral environment, you have all these inflammatory mediators that are actively communicating, whereas in the intratumoral environment, there's actually more associated with growth factors. And then we can kind of see that here, where red means more communication, blue means less communication. Interferon gamma is a very potent inflammatory regulator. So we can see that in the outside of these tumors is where all the inflammation is, but it's not actually on the inside. The important part about that is inflammation brings lymphocytes, brings an immune response, is indicative of some type of immune activity. And again, we're seeing that in the outside of the tumor, but not on the inside of the tumor. When we actually go and dig in a little bit further, one of the things we see is that when we look at just the totality of cell-cell communication, what we can see is that it's the periphery or the outside of the tumors that's actively most actively communicating. So these plots, essentially anything that's in the top right quadrant means that they're actively communicating, and they're color coded to match these spatial plots. And what you can see is that, there are these regions, these peritumoral regions are actually the most actively communicating regions within in the metastatic osteosarcomas, which we thought was really cool. We'll kind of skip over this for the moment and really go here. One of the things we also looked at is TGF-beta signaling, which is highly immunosuppressive. So if we're not having much inflammation on the inside and all the inflammation is on the outside, what's going on the inside of the tumor? So we looked at TGF-beta signaling, and this is a spatial plot. Then we looked at spatially derived ligand receptor interaction. So essentially what that means is who's talking to who, which ligands are talking to which receptors in these different spaces. And what you can appreciate from this is that it's a mess, right? It's just pretty much everywhere. So all different parts of these tumors are actually talking to each other. And one of the things we can do is we can start inferring activity. and asking questions, okay, not just looking at ligand receptor interactions, but actually looking at downstream signaling activity. And we can see that there is, just looking at TGF-beta 1 activity, it's actually pretty interesting that there's more so inside the tumor than on the outside. And then we can project the directionality of that signaling. And to be honest with you, this needs a little bit more work. We're still trying to figure out exactly how we want to tease this out, but it's just really showing that there's heterogeneity in TGF-beta signaling. One of the last things we'll go to in the sake of time, I just really wanted to spend time here. What I've showed this far is that there is a lot of there are a lot of similarities across the specimens, right? And remember, if we go back to the introduction, genomically, osteosarcomas are considered an N of 1 disease because they're so different at the genome level. But when we actually start looking at the transcriptional profiles and where those transcriptional profiles are actually located within the tissues, and we start asking questions about tissue structure, there's actually a lot of conservation. So when we integrated our samples and then did some clustering analysis, we actually saw that there is a decent amount of transcript. The transcriptional architecture is actually quite well conserved, meaning that gene expression profiles in one gene. Tissue-specific gene expression profiles are actually occupying similar regions of each one of these specimens. And we thought that that was actually kind of cool. So we wanted to really kind of capitalize on the conserved transcriptional architecture and ask questions about drug gene interactions. So one of the things we did is we took the genes that define each one of these clusters, we ran them through a drug gene interaction database, and we came up with a variety of drugs that target different regions of the tumor. So this is really interesting because this allows us to think about how can we best utilize combinatorial therapies, right? Rather than saying, okay, we're going to throw chemo A and chemo B and hope for the best, we can say, well, we want to target this specific region of the tumor and this specific region of the tumor because we know that these regions are really important. So can we actually utilize this spatial profiling to identify drugs that would target specific regions of the tumors based off of the genes that they're actually expressing? And this was just a proof of principle, but it actually worked out in the sense that some of the genes, some of the targets that we predicted are actually targets that have actually been used in osteosarcoma. So when we look at regorafenib and cabozantinib, I draw those out because those remarkably had, as single agents, actually prolonged event-free survival in patients with metastatic osteosarcoma. And we know that one of the aspects is that they target androgenic signaling, but they also target macrophages as well, based off the literature. And the regions that are actually highlighted are macrophage-rich regions of these tumors, suggesting that maybe regorafenib and cabozantinib are eliciting their effects by actually having specifically targeting macrophage-rich regions of the tumor as opposed to the angiogenic aspect of the tumors. So this gives us an opportunity to really learn a lot more from these clinical trials because we can utilize spatial profiling to ask questions about what have these drugs done to the architecture and structure and function of these tissues without actually having to ask for a ton of tissue. We just need a few slides. And that's important. You can see here the evidence of cabo and regorafenib showing some kind of efficacy in osteosarcoma. So the conclusions are, as we said before, but we'll kind of pick up where we left off. Inferred inflammatory and oncogenic signaling activities are compartmentalized. So inflammatory signaling in the periphery, oncogenic signaling inside, TGF-beta families signaling is pretty much heterogeneous and widespread. And the peritumoral environment is really highly active in communication, and there's a regionally distinct gene expression profiles that are actually conserved across specimens. So we're just going to really move forward and try to do this at higher resolution and really try to understand these three different aspects that were listed here on the right. And with that, I'd like to thank everyone involved. It took a lot of us to get to where we are, and we're really excited, but we're not stopping here. We're just going to continue to push this forward and make sure that we can do the best by all of our osteo warriors and osteo angels because we owe it to you guys. And with that, that's all I have.
Thank you so much. That was fascinating. And just
Should I
great.
stop sharing?
Oh, sure. Yeah, you can go ahead and stop sharing. Yeah. No, that was so interesting. And everyone, please feel free if you have any questions to add them to the Q&A, and we'll make sure we ask Dr. McEachron. But I like representing the remedial student population in the audience. I appreciated your summary point slides, but also as I was following along, I was kind of imagining in my head like this raucous, and I'm not speaking from personal experience here, but imagining this raucous party going on, with just like a bunch of bad stuff going down at this party, and kind of there are people standing outside trying to get in to like stop the party and like control
Honestly, that's the best way to think about it. So you can think about it a couple of different ways, but it's variations on a theme where, I'm not terribly old. I remember going to clubs when I was in college, right? A lot of interesting things happened inside those clubs, right? And the bouncers were there to make sure that only people who were going to contribute to whatever was going on inside the club were allowed in, right? Like all those lame people, they're like, yeah, no, you're not coming in. If you think about this metastatic osteosarcoma microenvironment, it's kind of like, for lack of a better term, the intratumoral microenvironment, the core of that tumor, that's the club. And you have this peripheral region that those are the bouncers. And those immune cells are trying to just really spoil whatever's going on. They're just trying to police and spoil what's going on. And the bouncer's saying, no, this isn't happening. Go to the club down the street, go to wherever you need to go, but you're not getting in. Or you can think about it another way in terms of real estate, right? We're talking about spatial profiling, location, right? That's what people say in real estate. Imagine that you have the tumor is a really interesting gated community, right? And it's a gated community because you want to exclude people that don't belong in that community. That's kind of what that metastatic environment is doing. It says, okay, I'm going to put up my gates, and this is a community for us. And unless you're going to come in and really contribute to what we have going on inside this little community, you're really not welcomed in. And so we're going to kind of stop you at the gates and not allow you to come in. Either way you look at it, there's something going on inside the tumor and something going on outside the tumor, and they're not cross-talking. So what is that barrier? That's where we want to try to figure out what that barrier is. And if we can figure out what it is, what it's doing, how it works, can we deconstruct that barrier? And does that allow immune cells to then come in? Alternatively, does it allow metastatic cells to come out, which would actually be horrible? So we're not sure exactly which way that's actually going to go. So we're trying to figure that out.
Oh yeah, that's a tough one. I didn't think about the coming out part.
Yeah. Yeah, there's two sides to the coin, and we just don't know what that's going to look like.
Yeah. Well, I would definitely be the one outside trying to spoil the party. And then
Yeah.
I liked the exhausted, the ones that can make it in are just exhausted from like
Yeah. They try so hard.
party.
They have nothing left.
And they just give up and they're like, fine, just be invisible. I don't care what's going on.
Yeah.
So basically trying to figure out, I mean, ideally it's like trying to co-opt it's like which part can best co-opt the bouncer to get them on their side.
Right.
So like,
exactly. Yeah.
Cool. Hey Walker, I think has a question.
Yeah, there was a question that came in the chat. They asked, is there a good way to mitigate the side effects of cabo? We have had 3 patients recently who have all had fatigue, extreme GI upset, skin ulcers, and eventually had to stop the drug after multiple dose decreases without tolerance.
I'm not a physician, so I can't really speak to that. I have nothing to say about that. I wish I knew more about the clinical efficacy and kind of how these things work in patients, but that's not my background of area of expertise. So unfortunately, I've got nothing for that.
You know what I would suggest, though, for the person who had that question is Turbo, which is our virtual tumor board. That's actually a place where people definitely discuss questions like that. So I'll respond and put some more information, but I'll just put in a plug for that. And then Daniel, do you want to take the next couple questions that came in?
Yeah, we have a question that says, should we be considering deep B cell therapy to disrupt the silencing outer layers of the tumors?
That's a great question. That's kind of what our data is leaning towards, or our data is suggesting that B cells are doing something. What they're doing, we don't actually know, so we have to really investigate that thoroughly to try to understand what's going on. The role of B cells in solid tumors is kind of emerging right now as we speak. That field is starting to grow, and it's really, really interesting data to follow. But we have a lot of work to do to figure out what B cells may or may not be doing in the context of solid tumors, and also what does that mean regionally? If there's B cells inside the tumor versus outside the tumor, what are they doing? Are they associated with tertiary lymphoid structures or whatever the case may be? So the data suggests that B cells are doing something, but we need to do a variety of functional experiments to really try to understand what it is that they're actually doing. But that's you're kind of on par with what we're thinking.
Okay, thank you. And another question, how about intratumoral immunotherapies instead of IV infusion therapies?
Yeah, that's a great question. So recently, a lot of people have thought about STING agonist as a really interesting therapy, and that will work for places that are easily accessible, right? So if you have melanoma or maybe certain types of breast cancer or certain things like that, where it's easy to have an intratumoral injection. But in osteo, there's a couple of things that we have to think about. One is the actual density of the tissue. You have malignant osteoid and chondroid. So essentially that's malformed bone. So it's not necessarily, depending on the extent of osteoid or chondroid matrix there, that might not be easily injectable, right? Because it's really, really, really difficult. And you have to think about questions of perfusion. The other thing is getting to some of those, if we're looking at this in the context of metastasis, depending on where those tumors are in the lung, that could also present a impediment in terms of being able to access them. I'm sure there's really, really smart people that are a lot smarter than I am who think about these things all the time and how to do intratumoral delivery. Um, I think that would be really cool. I just don't know how that would work, but it's also something that I don't spend a tremendous amount of time thinking about. So there very well may be avenues to pursue in that regard. But trying to elicit some type of immune response inside the tumor will be, I think, hold a lot of promise, however that may happen.
I wanted to follow up on you had a really interesting couple slides about, these kind of regionally like parts of the TME that were different and targeting different drugs and basically kind of testing different drugs against those different environments, and just kind of thinking through how that would translate eventually to clinic. Like, because I know a really common question is kind of which TKI to use for a
Yeah.
patient. And so as you kind of think through that, like how might that work? Because it still seems a little confusing to me, right? If there are all these
Yeah.
different, I mean, there are different regions within a tumor, and you want to kind of target all of them, don't you? So how do you do that without giving the patient like 5 different TKIs?
That's a great question. That's a really, really good question. And we do spend a lot of time trying to figure that out. So what we presented was pretty much a proof of principle that we can utilize this type of approach and this type of technology to potentially derive interesting therapeutic combinations that we can functionally test in the laboratory. I'm not saying anything about going directly to clinic. Um, One thing we have to say, and it's really important to think about this anytime anyone presents any type of kind of profiling data utilizing patient specimens, we're looking at a snapshot in time, right? We're only being able to infer information at the time at which that tissue was accessed. We don't know what's really happening before. We don't know what would happen after. All we have is that moment in time. So again, we have a lot of functional work to do to try to figure that out. I think that if we were able to understand what the different regions of these tissues are doing and how they're affecting the structure and function, then that might give us an opportunity to say, okay, well, maybe you need to treat with combinations A and B at this time point, and then maybe combination C and D at a different time point. But um, there's a lot of functional work that we need to do to really be able to try to figure out what that might look like. But again, this was proof of principle just to say, hey, can we utilize this approach to do something slightly different and maybe take a different look about how we might be able to think about combinatorial therapies? But there's a lot of work that still needs to be done.
And then just another question about just making sure I understand your points about the kind of preserved transcriptional architecture. So was that that it was across different samples that it was? Okay,
Yeah,
got it.
so so I guess rather than focusing on the malignant cells, the tumor cells, if we take a step back and not focus on the tumor cells, but focus on the body's response to the presence of disease, right? That gives us something that's a little bit easier to think about, and it's more conserved because we're not dealing with the genomic complexity and all these other things. We're just asking a question: how is the body responding to the presence of this disease? And what we're seeing is that that response to the presence of disease seems to be conserved. So if we move away from the actual tumor and trying to target and trying to understand how the body's response is being manipulated by the tumor to promote the growth of the tumor, that becomes an avenue that might be a little bit more broadly applicable. We can think about it like mosquitoes, right? You can sit there and try to kill every mosquito that you find, right? But it's not going to be worthwhile because there's always going to be another mosquito somewhere, right? It's kind of like the tumor cells. But if we understand the environment that the mosquitoes are in, we then just poison the environment, right? So that's actually how we deal with mosquitoes. We know that they need a water source. So you don't treat the mosquitoes, you actually just treat the water source. You treat what they need to survive. So if we take our attention off of the genomic complexity of disease and try to understand what is it about the tissue, what is it about the body's response, how do these tumor cells interact with the body's response, the tissue specific response to the presence of disease. What about that interaction are they actually dependent on? And if we could figure out what that is, then okay, well, what happens if we therapeutically intervene in that process? Does that give us a window of opportunity? Does that give us a new approach to thinking about how we might be able to tackle this? If it's that kind of peritumoral region, right? Those aren't actually tumor cells. Those are more associated with fibroblasts and this kind of wounding or scarring response. If we can get rid of those bouncers, if we can get rid of that, which is conserved across the specimens, does that give us an opportunity to get things into the tumor? So it's that type of approach, focusing more on the response that seems to be conserved as opposed to the genomic complexity that's going to vary between tumor.
Right, got it. Okay, so that helps me. Then one last question though about that. So like, does that response, does that change? Like, how does prior treatment change that? And also just the type of treatment, because I would imagine that maybe like chemotherapy versus molecularly targeted therapy versus immunotherapy might somehow change that over time.
That's a great question. The specimens that we utilize for profiling, all of those individuals had received standard of care MAP therapy. So in a treatment naive setting, we don't know what that looks like, but that's actually a great question. What does the molecular architecture of these tumors look like in a treatment naive patient versus a patient who's been heavily pretreated? Luckily, these are things that we can do in animal models and try to answer that question. And this is where actually utilizing canines becomes really, really important because they'll go through multiple rounds of chemotherapy. And the ability to harvest tissue from canines, we don't have the same restrictions that we have on humans. So we can get, depending on who you're working with, opportunities to get longitudinal sampling from canines at multiple time points throughout their therapeutic journey to ask those questions. How is chemotherapy actually affecting that interaction between the body and the tumor in a tissue specific way? And what type of selective pressures are there early on versus later? That allows, we can do that with the canines. We could also do it with mice as well. But it's utilizing canines in a comparative approach would probably be the best since that's a naturally occurring disease. So some of the complexity that we miss with mice, you'd still be able to capture there.
All right, well, we are out of time already, but thank you so much, Dr. McEachron, for joining us on OsteoBites today for really thought-provoking presentation. Really enjoyed it, and for making it better for pediatric cancer patients. More information on this and all OsteoBites can be found on our website at mibagents.org, on the MIB Agents YouTube channel, and at your favorite podcast place. We are going to be on a short hiatus for OsteoBites, but look out for a new episode of our Osteo AYA podcast, which is going to drop on October 17th with a Halloween theme, spooky sarcoma stories. So that will be in a few weeks. And then OsteoBites will return on October 24th. We are going to be joined by Dr. Joanne Toohey, who does work with our canine friends. She's an associate professor of surgical oncology at the Animal Cancer Care and Research Center at Virginia Tech and the Virginia Maryland College of Veterinary Medicine. And she's going to be joining us on OsteoBites to discuss her work studying histotripsy as a novel tumor ablation modality for treating osteosarcoma. You can find our OsteoBites lineup for the next few months on our website. And if you have any ideas for future topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. McEachron, Walker, and Daniel for spending an hour with us today, and for all of you for joining us. And we hope to see you back here in a few weeks on October 24th when we talk to Dr. Toohey. Thanks, everyone. Thanks so much, Dr. McEachron. Thanks, Walker.
Take care.
Thanks.